Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 11. The topic is Chromosomal Basis of Mendel's Law.
Discussing agenda for the topic is Introduction, Chromosomal Theory of Inheritance. Lecture outcome. After watching and listening this lecture, students will know about the similarities in behavior of chromosomes and Mendelian factors during meiosis. Introduction. Using improved techniques of microscopy, cytologists worked out the process of mitosis in 1875 and meiosis in the 1890s. Cells are small structures; they are not visible to the naked eye, and specialized devices called microscopes are used to study cells in greater details. So, with the passage of time, as sophisticated high-quality microscopes were discovered, the researchers, the cytologists, were able to study cells in more details. So we can say that the advancement in cytology is directly related with the advancement in microscopy. So when high quality advanced microscopes were discovered, the researchers were able to analyze the process of mitosis and meiosis. Sexual life cycles and gametogenesis was studied in detail. Gametogenesis is the process where the gametes are produced, the sperm and the ova. So the sexual life cycles were studied in deeper details too, and the behavior of the chromosomes during various stages of gametogenesis was analyzed. The predominant form of division through which gametes are produced is meiosis. Biologists began to see parallels between the behavior of Mendel's hereditary factors and chromosomes. Mendel performed his experiments with pea plant back in 1860s, and he concluded his findings and formulated them into two famous laws of genetics. The first law, law of segregation or law of dominance, which says that each trait is controlled by a pair of factors. Those factors stay together in a diploid organism, and during meiosis, during gamete formation in that diploid organism, these factors determining the trait are separated so that each factor enters one gamete and not both, means not both factors enters the same gamete. So this is the law of segregation. The law of independent assortment says that the alleles determining different traits are segregated into the gametes independently. It simply means that suppose there is an allele which determines the height of the plant, and that allele is dominant over another allele which is recessive, and there is another trait where an allele which is dominant determines green pot color. So the second law says that it is not necessary that an dominant allele for one trait will always enter into the same gamete with the dominant allele of another trait, and same is for the recessive, means the segregation is independent. The dominant allele of one trait can be a part of a gamete which has the recessive allele of another trait. So they studied the division in germ cells, in gametes, and what kind of behavior the chromosomes show there. You are already aware of that. You have already studied the process of meiosis, where there are two successive divisions that results in four haploid gamete cells. What happens in the beginning? The homologous chromosomes, they come close to each other, they align lengthwise in a process called synapses, which is aided by synaptonemal complex, and after synapses, the homologous chromosomes exchange segments with each other, and this process is known as crossing over by the cytologists and genetic recombination by the geneticists. So once the homologous chromosomes exchange parts with each other, then they are aligned on the metaphase plate, and after alignment the spindle fibers arises from both poles of the cell, and they spindle fibers attach with the kinetochore located on the centromeric region of the chromosomes, and as a result the homologous pair is separated from each other so that each homologue enters one gamete and no gamete has both homologues of a pair. So if you compare this process with what Mendel predicted after his experiments, is that the homologous chromosomes are showing the same behavior with Mendelian factors. Homologous chromosomes are two, it's a pair. Mendelian factors also exist in pairs. Mendelian factors are there, both the members are there in a single diploid individual, and during gamete formation, the two factors are separated so that each gamete has only one and not both, and same is the case with the chromosomes. The homologous chromosomes are there in a diploid individual, but at the time of gamete formation, the two members of a homologous pair are separated from each other so that each gamete has only one homologue of the pair and not both. And once the process of fertilization occurs, the pairs responsible for determining a trait is restored, means gametes are haploid. In spermatogenesis, sperms are produced which are haploid. In gametogenesis, the ova are produced which are haploid, and when these two haploid germ cells combine with each other, the diploid number is restored. For example, in humans, 23 chromosomes are present in the sperm, 23 are present in the normal ovum. So when they fertilize each other, the zygote becomes diploid and has 46 chromosomes. And the same statement applies to Mendelian factors. The diploid number is restored at the time of fertilization. For example, the sperm cell from your father has a particular allele for determining the skin color. Similarly, the ovum, the egg cell from the mother, also has the allele which determines the skin color. Now the alleles may be same or they may be different from each other. So when the fertilization takes place, both the alleles are reunited with each other. So around 1902, Walter S. Sutton and Theodore Boveri and others noted that these parallels, and they began to develop the chromosomal theory of inheritance. So initially, Mendel's Mendel's laws concluded that these factors are involved in heredity, but once the behavior of chromosomes was studied, then it became evident, it became clear that the chromosomes are exhibiting the same behavior as that of the Mendelian factors, so they also have a role in heredity. The chromosomes also have a role in heredity in determining the physical attributes of an organism. So now chromosome theory of inheritance. According to this chromosome theory of inheritance, Mendelian genes have specific loci position along chromosomes. So this theory also postulated that the Mendelian factors which we now call as genes are located on chromosomes, and it is the chromosome that undergoes segregation and independent assortment. Means the genes are located on the chromosomes, and the chromosomes are the vehicles which carry genes. They provide a seat to the gene, and the behavior the chromosomes shows can be attributed to the behavior of Mendelian factors. The separation of homologs during anaphase 1 accounts for the segregation of the two alleles of a gene into separate gametes, and the random arrangement of chromosome pairs at metaphase 1 accounts for independent assortment of the alleles for two or more genes located on different homologous pairs. Now here is diagrammatic representation of what we already studied in the text. So this is basically a dihybrid cross in which the individual differing for two traits are crossed with each other. The one of the parents has round yellow round seeds, which is a dominant trait, and the other parent has green wrinkled seeds, which is a recessive trait. So one of the parent is homozygous dominant for seed color and seed shape, the other parent is homozygous recessive for seed color and seed shape. Now, because those parents are diploid, their chromosome number is 2n. Now gametogenesis takes place, they produces gametes. So because it is a true breeding variety, so all the gametes produced will be of the same type. Similar is the case here, it's a true breeding variety, so all gametes will be of the same type. Now the two gametes fuses with each other through the process of fertilization, and this becomes the F1 hybrids, F1 generation. Now you can see that the F1 generation, there is a dominant allele for seed color, and there is also a recessive allele for seed color, and similarly one allele is dominant for seed shape and the other is recessive for seed shape. Now here the basic concept starts from here. So these are the F1 hybrids. Now some of the F1 hybrids will be males, some will be female, but they are heterozygous, and they are exhibiting the characteristics of the dominant parent. Now the F1 hybrids will also undergo gametogenesis where gametes will be produced, and here you can see that the gametes produced are some of the gametes are carrying the dominant copy for seed shape and the recessive copy for seed uh, dominant copy for seed shape and recessive copies for seed color, and the other gamete is carrying the recessive, one copy is recessive here, the other copy is dominant. So this explains that the segregation is independent, means the dominant allele of one trait is not always the part of dominant allele of another trait. This segregation can be independent. You see here the capital R and capital Y, small r, small y. Here the small r, capital Y, capital R, small y. So the grouping is independent, and then the process of meiosis takes place, and the homologous chromosomes pair lengthwise with each other, and then they are separated from each other. So these are the gametes which are finally produced. You can see all kinds of combination is there. Some of the gametes are having dominant copies for both traits. Some of the gametes are having recessive copies for both traits, and some of the gametes are heterozygous. They are carrying one dominant copy and one recessive copy. So all kinds of allele combination is possible. You can see here those gametes has dominant copies for both traits. Those has recessive copies for both traits, and those are heterozygous. One copy is dominant, the other is recessive. So this is independent assortment, means the alleles determining different traits are assorted independently. If it was dependent assortment, then the dominant allele of one trait would have always be along with the dominant allele of another trait. But in independent assortment, all kinds of allele combination is possible. Here we have slightly magnified the image you saw in the previous slides. These are the two parents, true breeding. One is true breeding and homozygous dominant for seed color and seed shape. The other is also true breeding variety, homozygous recessive for seed shape and seed color. Then they produces gametes because they are homozygous true breeding varieties, so all the gametes will be of the same type. Then here the F1 generation process of gametogenesis starts in the F1 generation, and now the gametes that will be produced, the alleles will be segregated independently. You can see these are two alleles for determining the seed color, seed shape. These are two alleles for determining the seed color. Seed color, yes, this one is the seed shape, round. It is dominant against recessive, dominant against recessive, recessive against dominant, dominant against recessive. So all kinds of allele combination exist here. Now they will produce gametes. Now you analyze gametes, both alleles dominant, both recessive, heterozygous, heterozygous. So all kinds of allele combination is possible. This is in case of independent assortment. So I have put figures. Some of the figures are without explanation, some have explanation, so that you can understand it for yourself. And this is basically the different forms of the cross that we analyzed in the very first figure. Thank you very much.